So Sandeep and I are going to try something where both of us will together give this this lecture and the next lecture. What we're going to try to do in this part of the our talks is start with a biological phenomenon which we've been studying in my lab and so on for some time. Try to reduce that biological phenomenon to a question that can be addressed using mathematical modeling and physical concepts, and then try to see how these two can be integrated to get some insight into a biological phenomenon. And of course, this is related to the areas we talked about yesterday. Okay, so the biological question here is coming from a fairly old question in classical cell biology and the cell cycle. You have cells. The normal cell goes through a classical cell division cycle, right? So you have G1, S, G2, M phases in the classical cell division cycle, and this is how classically mitotic division and proliferation takes place. And in each part of the cell cycle, experimentalists and could systematically work out what is going on. So you have an initial growth and synthesis phase where you're at this point making all the DNA that you need to go through a G2 phase, and then you have a replication which results in finally two cells being made. So this is the classical cell division cycle. But we also know that, so this is for the state of a cell which is proliferating. We understand this really, really well. But in addition, you have another phase which is G0 or quiescence phase. Now the biological definition of quiescence is very, very ambiguous. Depending on whom you ask, they'll say this is what quiescence means. But practically, what it means is a non-dividing stage. So the cells at this stage will not carry out DNA replication and will not divide into two cells. That's really the functional definition. But if you ask different cell biologists, especially people in studying quiescence at different levels, they'll argue violently over what that means. It could be everything from an ametabolic stage, which is truly quiescent, which means nothing is happening, to something that's almost dead, and then there'll be an argument between senescence and quiescence, something there, or something that's at this cusp of reversibility. But what we do know is that many cells can go to an non-dividing state, G0, and then with the right kind of cues, exit G0 and come back, re-enter G1. So that's re-entering the cell cycle, classical cell cycle, okay? So as a phenomenon, there are various ways that as biologists we document all these processes. But it's still hard for us to explain what it takes for a cell to either go from G0 and come back to G1, re-enter the cell cycle, or exit the classical cell cycle, get out of M phase, and go back to G0 and remain in a quiescent phase. Okay? Now, separately, so this is the big backdrop that we're going to try to address in this particular story. Now we'll come to the specifics of what the biological phenomena that we were studying, and where it looked like we might be able to address some aspects of this bigger cell biology question. So this comes to a phenomenon observed in yeast many years ago. So the experimental observation was as follows. You have yeast in a chemostat. So a chemostat is just a way to grow cells to very high density, but maintaining constant nutrient flow, aeration, oxygenation, pH, and all of that. And here, when you have cells at high density, these are just yeast cells grown to very high cell density, treated as a batch culture, which means you're not feeding too much. You're treating it first as a fixed state where there's nutrients in there in the media. Yeast keep growing, use up all the nutrients, and then they start starving. And when you do this, you can observe an oxygen profile. So the measurement here is just a readout of oxygen consumption or oxygen in the media. That's this. You see that oxygen in the media keeps decreasing. So this is dissolved oxygen. At this point, all the nutrients are used up. The cells are at very high cell density. So then they go back to this kind of starving phase. And this phenomenon then, what was observed was that now at this point, if there was a small amount of steady state nutrients being provided, the entire culture population of millions and millions of cells spontaneously went into these kind of oscillations. And these are very robust oscillations that can go on for days and days and days and days, as long as a small amount of nutrients was steadily feeding in at a fixed rate. Okay? So this was actually observed initially by people in the brewing industry who had started using chemostats to make large quantities of beer or wine and so on. And then experimentalists in the lab were obviously a little late to see this, because usually the biotechnology industry is way ahead of this, way ahead of us. But once we stumbled on this, there were a lot of interesting things we could look at, because what we were seeing here was a large-scale synchronized oscillation of cells behaving in some kind of synchronous fashion. So several groups studied this extensively, but one of the most elaborate systematic studies was done by Ben, Tu, and Steve several years ago, so 2005. And what they first started doing was collect time points all across each oscillation and just looked at gene expression profiles to see if there was something happening at a bulk population level. This was bulk data. What they saw was really, really striking. They essentially found about two-thirds of the entire yeast genome, all the genes in yeast, showing highly periodic synchronized oscillations, which seemed to have some pattern. So this was the global pattern of gene expression. So it was about 60% of all genes in the genome, showing these oscillations across these cycles. And just for your information, one cycle length from here to here, depending on the system you're using, is between 2.5 and 4 hours. Okay. Now, when they tried to bin these groups of gene expression profiles into functional classes, what they then saw was much more striking. Okay. So this is what Ben saw some years ago. You could nicely break down these oscillations into 3 very distinct functional groups. So in this phase where you're rapidly increasing oxygen consumption, you had all genes related to growth really peaking. So the kinds of genes you would see here are things like ribosomal genes, rRNA genes, tRNA genes, sulfur metabolism, amino acid biosynthesis. This is what you would say is a classical growth output for a cell. You see a huge increase in expression in this phase. Immediately following that, in this phase, where there is now no longer high oxygen consumption means you're decreasing oxygen. Here you're stopping oxygen consumption and going back up. You see something else happening. This is where the classic DNA replication cell cycle genes came up. So you'll have all your classic cell division machinery and everything going up over here. You'd complete cell division over here. And then you'd reach this long phase, about 2.5 hours, where you had a gene expression profile consistent with what you'd call a semi-starving quiescent type of a cell. All the classic markers of quiescence, autophagy, heat shock proteins, something about the ubiquitin proteasome system, all of them peaked over here. You could see these 3 distinct phases as a measure of gene expression. And so this, they could break it down and call it a yeast metabolic cycle, because primarily you're seeing metabolic oscillations and gene expression profiles that seem to segment the life of a simple, any cell. In this case, they called it the life of eukaryotic cell, but this is what you'd go through. A high growth phase followed by cell division, followed by a phase of non-growth quiescence or whatever. This they could also then systematically, this is Ben's group along with a couple of other groups, systematically looked at metabolite oscillations in this whole cycle. Again, they could see very nice patterns, very consistent with the kinds of things you would predict over here. So you would see a very nice profile or grouping of metabolic oscillations in these oxygen oscillations. So you'd see in this phase things coupled to growth, a whole bunch of things going up here. The next thing here you see are nucleotides and other things that you require during cell division going up and peaking and being consumed, and then it's going down. And here you're building up a whole range of carbohydrate storage, carbohydrates and amino acids, which you would then use to re-enter growth. So beautiful. So we have this entire metabolic life of the cell being portrayed here in this huge bulk population. But, and there's a little more, and this is where the little details start coming in. So this is your oscillation, right? What we see is the synchronized behavior of all cells, but within this, there was a very nice little bit of heterogeneity coming in. First is that not all cells entered cell division. So this is cell division that happens over here as you're going up. And in this population, somewhere around 35% of the cells would divide. That's it. The remaining 60 to 65% do not divide in this population. Second, in this place, 100% of the cells over here are non-dividing. So there is some kind of a heterogeneity present in this system. And even though the bulk data clearly, the bulk gene expression and the bulk metabolite data clearly indicated multiple, two distinct phases, this growth phase and the division phase. During this phase, it looked like not all cells are dividing, only 30% of the cells are dividing. So at least we could experimentally ask if all the cells that entered this phase completed division, and this we could clearly see that the only cells that go on to complete division are the ones that commit to this entire phase, this growth phase, and you could do this using a set of gene expression markers or something like that, reporters that you could then fax out of the cells and so on and so forth. So you could clearly show that the cells that had all these markers highly expressed were the only ones that completed cell division, all the others wouldn't. So essentially, so this was how me and others as biologists were dealing with the system. We were using bulk data, bulk information to make assumptions about what a cell or what a cell does during growth or division and so on. We were also using the metabolic cycle to understand the function of unknown genes, looking at gene expression. Suddenly, if you find an unknown, uncharacterized protein whose transcript increased in this growth phase, we could predict that it would be a growth-related gene and so on and characterize it and so forth. But the low-level models that we as biologists were making were something like this. We would put something like, yeah, there's a growth phase, there's some commitment, some decision, we have no idea what's going on, but a fraction of the cells divide. This is just showing you how you can see this division. In yeast, you have budded cells, so you can see budding over here, and you can count the budded cells. It's only over here, and only about 35% divide. Okay. So this is the system that we had. So now though, if you want to think about this phenomenon itself, what you can clearly see is that there is emergent heterogeneity. The whole system is a stable oscillation because these oscillations can go on endlessly, which means there's a switching between growth, non-growth, growth, non-growth, growth, non-growth, right? If you want to break it down into a physical kind of a problem, you might be able to view this as a bistable system. Because if you're going through these oscillations, even though I said there are 3 bins for gene expression profiles, you might be able to reduce this to 2 general phenomena. One is all sets of processes related to quiescence, where cells are basically in a non-dividing state. So I'll use the loosest term of quiescence, basically saying they're non-dividing, so they're quiescent or quiescent-like. And the second phase, which is this entire dip and going up, where all the processes related to growth, and because the growth is so closely coupled to division, cell division, both of them are coupled. So you can view this as another separate bin. So what it looks like to the eye is some kind of a simple bistability. So instead of treating these as multiple gene expression profiles, we try to reduce this to a very simple, apparently bistable system with 2 states, a Q state and a G state. And what's happening is that there's some decision happening to switch between Q and G, but as soon as G is complete, you're going back to Q. So you're resulting in a continuous steady oscillation. And so now if you're thinking of this kind of a system, you can try to ask how might such an oscillator be developed with the general idea that if we figure this out, we might be able to say something general about how cells can toggle between quiescence and growth, growth and quiescence. So then we reduced this apparent bistability to 2 or 3 possible biologically plausible scenarios. So now that we know that this is a nutrient-dependent oscillation, this is a bona fide metabolic cycle, thanks to all the metabolic data that we have, we came up with these 3 plausible scenarios based on the kinds of systems that you have for heterogeneity out in biology. So there's actually okay, this is a chemostat system. So there's a little detail that I'll tell you now. So this is a I'll just clarify this extensively. This is a high density culture. The cell number, the cell OD here is about constant, so it's about OD of 15. And the reason it's constant is because there's a chemostat, so there's a small feed, so there's also a small outflow. Okay, so we can maintain cell density despite this growing and so on and so forth, because the feeding rate and the outflow rate has been standardized. Okay, the amount of cell death in here is actually relatively very low. So if you measure at any point of time, you may see about 5 to 10% of the cells that are showing up as dead, and that's it. So it's not a very high cell death rate. We have not accounted for cell death in the model that we're going to build over here. But that's the rate over there, 5 to 10% that's going on. No. So yeah, and also the cell death. So, even though this is the oscillation and growth and division is here and autophagy quiescence is here, if you measure cell death, it's fairly stable across. So it's vaguely in that 5 to 10% and not a big oscillation over there. Okay, so these are the scenarios that we came about that might be biologically sensible. You could have a stable heterogeneous system where one set of cells is producing a common resource, like we talked about yesterday, that's being taken up by only another set of cells, which is a user. That's the only one that's dividing. It means now you're separated into a dividing and a non-dividing type population. The other possibility is something like you'd see in a quorum sensing scenario. All the cells put out some non-consumable resource. This will build up, but only some cells will respond to that and carry out division, while others won't respond to that and remain, or respond differently to that and remain non-dividing. And the third scenario is a variation of this, which is that there is some metabolic resource that's being produced and consumed, presumably by all cells, but the amount of the metabolic resource present within the cell will decide whether you divide or not. So these are the 3 broad scenarios that we put out that we could now systematically test. And this is when Sandeep will start talking more about the model. But vaguely then, if we had to take any of these scenarios, these are the parameters we would want to put in. Just the 2 states, a Q state and a G state. Some resource Q or alpha that's in the system, and we won't define the resource very tightly initially. There's a parameter for production and sensing and utilization. And primarily we'll be looking at growth rate, switching rate, which is from Q to G or G to Q, and the fraction of cells in any one state. You can either take the Q state or the G state, it won't matter too much. So this is how we went about starting to build the model. And Sandeep will now start walking through the entire process of how we built the model. Just one little detail here. The shape of the oscillation is slightly variable depending on the strain of yeast that you use. In all cases, you have these general oscillations, but there are slight differences in patterns. In all cases, cell division is in this going up fast rise phase, but there are slight differences in the way they drop. You might have a fast drop and a slow drop, or a fast drop and a slow drop with a little bump, or a very fast drop and only a bump, or a fast drop and a slow rise and then a fast rise. Okay, but in the end, cell division is over here. All of these scenarios have the quiescent phase, so we could still remain with this idea of binning this as a G phase and this as a Q phase. And now Sandeep will just walk us through what we did to build the model, and then I'll come back after he builds the model, maybe in the beginning of the next session, and see how it makes sense biologically or metabolically. So that becomes kind of the question, right? And we'll address this question again maybe after the model is built, because now I think we understand. So this is part of the question. We didn't know. So all the things we did know were as follows. Let me put this down. This is useful. How can you explain Q to G switching? Right? From this set kind of, how many were oscillating? We're only 50% oscillating or not, so that's one of the scenarios that we could put in over there. What can control oscillation? And then subsets of these questions are same cell dividing in each oscillation, as you have multiple oscillations, versus different cells in each oscillation. All of these are variations of this broader question of how to go from Q to G, but all of these we actually could not understand just by looking at the bulk data that we had, right? We cannot easily say that, and then once you take out a cell from the system, it is no longer in the system, so that it no longer oscillates. So we cannot then manipulate and go on and do something else in the lab. All we can do is take cells at any population, which is bulk, and get some overall readout for that bulk population. Or we could take it out at one point and sort and get two populations, and then say what is happening at that point of time, but it doesn't tell us anything about what's happening earlier or what is going to happen next. And we can't put it back in the system. There's no experimental way now where we could intelligently label some cells and see what's happening in each cycle and so on and so forth. This is why we really need some kind of a model to clarify our thinking over here. And we'll come back to more questions after Sandeep builds the entire model, okay? So, yeah. Any more simple questions now on the biology or the phenomenon itself? Okay, so we can wait till the model is built. Good.
So I am going, as Sunil said, I'm going to walk you through how we built a model for this, but simultaneously in the process of that, I thought it would be interesting for you to maybe see a little bit of how a collaboration between a biologist and a theorist works, how the thought processes go. And as you, in analogy a little bit with what's going to happen in this lecture, you'll find that one of the things that happens is for fairly long periods of time, we go off and do our own thing. You know, if we were swapping every 5 minutes, you'd all get confused in this lecture. So I'm going to talk now for pretty much the rest of this lecture and maybe a bit of the beginning. And that's also a bit reminiscent of how it works. I got some things from listening to Sunil, and then I went off in some direction. And then after a while, after doing some stuff, I came back, talked to him, and then, you know, changed course, he changed course. So, you know, you've heard a fair amount of details about the system, what kinds of experiments are happening. I think I had heard this talk from him once, more or less the same thing, perhaps a few less details. And I had perhaps had one conversation with him before I got excited about all this. And he's written down a bunch of questions that he sort of thought of. I'll put down next to it what I got from talking to him in those initial conversations. And you'll see that in some ways I abstracted out certain features and I forgot about a lot of other details. So what did I get from Sunil, more or less from the lecture you heard here? I got a few things. One is that there are two states. So that was kind of, and there's a Q and a G, and roughly speaking, one has proliferation, one is a non-dividing state. The other thing I got is that in a chemostat, even with the total number of cells, so let's say the total number of cells is being constant, you can still get oscillations between these two states. from Q to G or G to Q and back. So in some sense, I immediately forgot about all the stuff about dissolved oxygen and all that. I was not thinking of these oscillations as being whatever he was actually measuring. I very quickly abstracted that out. And I was thinking of the oscillations as really being the fraction, as he said, the fraction of Q cells. And if you remember, he sort of said something like, you're close to 100%. And then something happens, and he had all these, he had these various curves. You can't see the shape of the curves, but there are different sorts of curves in different strains. But in some sense, there was a sort of a flat period. There was a fast drop, and then various different shapes happened here, but one possible thing could be a little bit of a rise, then a fast rise, and something like this. And this level was around 30, he said that 35% or so. I'm sure that varied from cell to cell, but you had something like 35 to 40% or 50% cells. Sorry, 35 to 50 were growing. So this was more like 65%, let's say. So I was thinking of oscillations of this sort. And one thing that immediately came to mind, just looking at those shapes, and in fact, all of those shapes he draws, is that there were two time scales. So let me put that down. That's the third element that really struck me. There were a separation of time scales. So there are things that are happening slowly over a period of, say, 2 hours. There is transitions that are happening much faster over a period of, say, 15, 20 minutes. So you have these large drops from the quiescent, mostly quiescent stage to many more dividing and growing. And then some other slow process happening over here, which is also perhaps around 1.5 to 2 hours. And then again, something happening rather rapidly. So 2 time scales was very apparent to me. And this was enough. At this point, I think I almost stopped listening to Sunil because he was talking about various metabolic stuff and so on, and it was getting too complicated. But this was enough to excite me because I knew from some earlier work I had done how to take a bistable system and produce an oscillator, create an oscillator by adding some processes, which would exactly have 2 time scales. And I was fairly confident I could produce shapes like this. So what I'll do in the next, say, 15, 20 minutes is walk you through that idea. So what happened, so roughly chronologically speaking, is at that point I went off and started doing all this and started thinking about how I would take the ideas I had of taking a bistable system and creating an oscillator out of it, which had 2 time scales, and whether I could adapt that idea to the yeast scenario, whether I could sort of take some of the processes I know would be happening in yeast, the switching between cell states, the growing, the proliferation, and could I adapt the idea I had of making an oscillator from a bistable system to the yeast? Okay, so and then only then did I come back to Sunil at some point and say, oh, here's stuff I can do, here's things we can do, and we started talking about what that means for the yeast system. So for a while, paths diverge and then come back and diverge and then come back. And I think that's very common in the way theorists and experimentalists interact. So let me come to this idea of making an oscillator starting from initially a bistable system. And we'll use some of the ideas I talked about last time. And I'll give a small recap of some of the bistability ideas, which I'll need. So the idea was something that we had called frustrated bistability. And the idea was simply to take a bistable system and somehow destabilize it so that it had 2 states, but it could never really stay in any one state for very long before jumping into the next. And I'll tell you how we do that. But for those of you who are familiar with oscillators and different types of oscillators, I should say this is just a subset of a set of oscillators which are called relaxation oscillators. So if you're familiar with relaxation oscillators, you will see pretty quickly where I'm going with this. But for those of you who don't know what relaxation oscillators are, it doesn't really matter. These are just terms. Okay, so let's start go back to what we did in my last lecture and start with a simple bistable system. So you remember we had a single protein activating itself. And I had said that you could write down something like this, where there was a production term and a degradation term. And the production term, because I've kind of defined this as an activator, should be a function that does not decrease. Should be an increasing function of some sort. You remember this? People are sort of still remember what I did last time? Okay, so let's imagine, let's take a particular case just to settle the intuition. Let's imagine I had, I'm going to parameterize this in some way, but I'm going to write down a hill function of a sort. This is just to give you the intuition of what I'm going to do. Don't worry about the specifics, it's just a choice I'm making. So imagine a thing like this. You remember we had drawn as a function of A, we drew, so this is A, this is this part, and then we would have to draw this part. This function is a sigmoidal function. It's a hill function just like the ones we were looking at in the last class. It starts off, so when A is small, the resultant value is alpha times beta. Let's assume beta is very small. Let's say that's something like 10 to the minus 3 or something like that, some very small leak. So when there is no A, there is a small rate of production. When A is very large, then the rate of production is alpha. So this kind of a function and goes something like that, where it starts at some alpha beta and it goes up to alpha. Okay? And if you've chosen the values and so on correctly, and alphas and betas and so on, you can get it to cut 3 times. And if you remember, this is what we had discussed. You can work out the stabilities of all these steady states. And this one will be stable, this will be unstable, and this will be stable. And that's simply because here the production, the degradation rate is larger than the production. So if you're on this side of the steady state, you will come back to it. The same thing on that side, you will come back to the steady state. So when you have the stable steady states, whichever way you perturb, you come back. In the unstable state, whichever way you perturb, you get pushed away. So you have a bistable system if you choose your values alpha, beta, and so on correctly. But of course, so what I want to do and go forward from what we had done in the last class is to actually ask what happens as you change these parameters. Your bistability may disappear, and indeed it will. So let's examine how things change as you change alpha. Okay? So imagine now you started with some value of alpha, and now you increased it. Let's say you doubled it. What will happen to this curve? This curve remains the same. There's no alpha over here. So only thing that will change is this. And I picked alpha because it's a very simple way it affects the whole thing. It just multiplies the whole curve, right? So all that's going to happen is the curve is going to go up. You are going to multiply all these values if you increase alpha. So you will go up. So this is some curve with some value of alpha. And the curve that you will get with another value of alpha, let's say a double, could be something like that. Does that make sense? I mean, maybe I'm not drawing this very well, but imagine you're doubling the value and doubling the value there. So this should probably go up a lot more. So if you double the value, you would actually go up a fair amount. But the point is still that you could get a system which no longer cuts in 3 places, but only cuts in 1 place. And let's think about the stability of that one place now. This is a stable fixed point. And you can check again using the same rule that we had. Here the degradation is larger than the production, so things will go left. And if you perturb on this side, it will go that way. So you can actually, by changing alpha, you can move from having a bistable system to a monostable system. What if we decreased alpha? Well, eventually, if you decrease it enough, you could probably get something that comes low enough that it falls below that curve. Again, you are in a state where you would only cut in 1 place, but that place is now down here. and not up there. So you can see that by adjusting your alpha, you can firstly remove the bistability, but also in removing the bistability, which of your stable states survives, so to speak, is different. So let's try and draw this out, because this is the heart of the idea that I will use to make an oscillator. So I want to draw a different sort of, I want to draw a graph of alpha on the x-axis. And here, what are the steady states that are possible in A? What are the steady states possible? So when alpha is 0, this doesn't exist. And all you're going to get is A equals 0. So you know you start here. You start somewhere there. You know that when alpha is small enough that this whole curve lies below that line, then there's only going to be one stable state, and it's going to be low. And now imagine this alpha starting very low and slowly going up, but not much. Still keeping that state. That state will move a little bit along this line, right? It'll increase. So if I plotted those steady states, I would slowly see some kind of increase. And this state would last all the way until I got to a point where I was somewhere here and just cutting the line. Yeah? All along, these states would remain, and now it would disappear. So at some point, at some alpha value, this state would no longer exist anymore. That's it. It doesn't exist. There is no state here. What states do exist once you jump over there? Well, clearly at that point, a high state does exist, because this was the line, and it cut somewhere up here. So this state exists. Okay? But in the bistable region, even below that, this state exists and that state exists. So there is some region of alpha where the high state was also there. So this is what the curve would look like. If you're only plotting the stable steady states, you would find for alpha, as you start from 0, there would be a low state only for a while. Then at some point, at some critical alpha, you would start cutting in 3 places. And when you cut in 3 places, you would suddenly have a high state also show up. I mean, I've probably not drawn this very properly. It actually probably comes down quite a bit, like probably something like that. But there is a point at which you suddenly hit bistability, and as you increase alpha, you keep going until again, now, this curve goes beyond the line, and you again have only one state, but it's only the high state. So this is a region of parameters where you have bistability. Above this parameter value, you only have the high state. Below the parameter value, you only have the high state. If I also drew the unstable state, it would actually be, well, let me draw this a little bit more reminiscent of a plot that Sunil drew yesterday. He showed in his thing, he actually had drawn a dotted line over here. That just indicates the unstable states. I don't know if you remember from his slides that he had a curve which was going like that. That was exactly this curve. As a parameter is changing, what are the steady states you can get? You get 2 stable steady states in a certain region of parameters. And that's very typical of bistability. There is a certain range of various parameters. You could do this with other parameters. You could do it with this. I could have put a half-life over here and done the same thing with the half-life. And you can imagine, instead of changing, if I was changing the half-life, instead of changing these curves, I would be changing the straight line. I would be making it steeper or shallower. And the same thing would happen. If it is shallow enough, you only get the low state. If it's steep, you only get the high state. And in between, it cuts in 3 places. So that's the essential point. We have to remember that as you change a parameter, there'll be a region of monostability, then bistability, then monostability. And this is what I'm going to exploit in order to make an oscillator. So now think about, as I build the oscillator, think about, we were drawing arrows, right, in various places to show how the flow would be, how you would get to the steady state if you started away from it. In this case, alpha, beta are just constants. They are just parameters. So they are fixed. So if I was to try and draw arrows to try and describe how the flows are here, basically, if I wanted to know what happens if I start with this alpha, but a small value of A, essentially, I can sort of draw arrows like this, which will all be vertical and show that you will end up at that stable state. If you started above, you would come down. And here again, the arrows would be like this. Here, the arrows would be like that. And here, the arrows would go the other way. If you're on the wrong side of the other side of the unstable state, you go down. So these are the stable states. And if you were here, you would go all the way down here. So these are the flows, given that your parameters are just constants. And here's the trick that I'm going to do. In order to build an oscillator, I'm now going to add something to this system, add a process, which makes the parameter, which was a constant before, no longer a constant, but dependent on the state of the system. So I'm going to make this another variable, another dynamical variable. So I'm going to change it from a one-dimensional system to a two-dimensional system. And there are particular ways you can do this in order to get an oscillator, and especially an oscillator that has two time scales. So here's the trick I'm going to do. So let me call this another variable. So instead of B, I'm going to call this, instead of alpha, I'm going to call it B. And I will write down a differential equation for this. And it will have the following property, that A will inhibit B, and B is activating A, effectively. So I will have something like sigma 1 plus A by some K with some Hill coefficient minus some gamma B. Very similar to the kinds of equations we wrote last time. I'm imagining that A was a transcription factor that was activating itself. It was binding somewhere to its own, near its own gene, and helping a polymerase come and express that gene. Now I'm saying it also has another binding site at another gene, at another protein, at another gene, B, and over there when it binds, it inhibits the expression of that protein. And here's where I will put, so is this clear? I'm changing the system now. I had a bistable system initially, and I'm now adding something. And I've just added something that effectively makes one of the parameters of the bistable system no longer a constant, but under control of the variable itself. And here's where I'm going to put in 2 time scales. I'm going to put in 2 time scales by demanding that sigma and gamma are very small. Maybe I should mention that, you know, we took an equation like this with alpha, beta. If beta is around 10 to the minus 3, and I made a choice of gamma equals 1, over here, the rough numbers where you get this are around 2, maybe I think it goes up to about 5. So it's all of order 1. All the processes happening, this is 1, this would be around 2, 1, 2, 5, 6. Still everything order 1. In other words, the time it takes when you start here to get there would be in these units, 1 or 2 time steps in these units. I'm demanding that these are much slower, much, much less than 1, which means whatever processes cause B to change are much slower than whatever was causing A to change. Okay? So what's the consequence of putting something like this? So imagine this is now a lot smaller than 1, and let's come back to looking at the flows here. Now, instead of having a constant alpha, I've changed this to B. This is now a dynamical variable, and the flows will depend. The arrows will now start bending. They won't be vertical anymore, because there is a certain dB by dt which is non-zero, and which depends on A. So the arrows now have to start bending away from the vertical. But if I take this huge separation of time scales, it's not going to bend very much. That's the key. If these flows are most vertical, in this new system where I've introduced a new variable, but I've said that processes that change that variable are very slow, you know that these can't bend very much. They'll bend a little bit. So let me redraw them. Maybe they'll bend slightly, but they will largely look very similar. We'll have to see which way they bend. We'll have to figure out, but you can make out that they're not going to bend very much. So they're largely going to be still mostly vertical. So let's now imagine in that scenario where the arrows are still mostly vertical, what kind of trajectories you can get if you start somewhere. So imagine I started here. Very quickly, I would go up, and I would come very close to this steady state. This former steady state of the previous system, which was a bistable system. I would come somewhere there. Once I'm here, notice that on this line, this was the line which was a steady state of this equation, right? So along that line, dA by dt has to be very close to 0. You remember in the 2D systems we drew for C1 and Crow, we drew lines which were d Crow by dt is 0, the C1 by dt is 0. This entire curve is the curve dA by dt equals 0. Which means once you're very close to the line or on the line, you're not changing A very much. That makes sense with the flows, right? The flows are all pulling you towards this line. So what's going to happen once you get there? And that's the critical thing. That's where the oscillator will show up. Once you get on this line, you're not changing A very much. But A has some value, a high value, and A is inhibiting B. So let's assume this equation, the way I've written it, is going to make B start to decrease a little, but very slowly. So you will be sitting on this line, and then B will start to go down, and you'll come to a new point on this line, and then it'll go down further and further and further, and you will start creeping along this branch. Okay? Is that clear? Does that make sense to everyone? You understand why you'll very quickly jump to this line and then start moving that way? If you set up the parameters correctly, you can always make this happen. You can start creeping down the line because along the line, A is large, it's inhibiting B, so B is going to start decreasing, which means that direction. And it'll keep decreasing until it reaches this point, at which point it'll decrease a little bit more, and then it sort of falls off the edge. And when it falls off the edge, it's a little beyond here, and what are the arrows going to do once it falls off the edge? They will really rapidly make it fall down to the bottom branch. So suddenly A will drop, B will not change very much, very quickly A will drop, and then A is now low, and the inhibition is lifted if you have set up the parameters correctly. And once the inhibition is lifted and A is low, then you're on this branch, and you will start to creep up. So B will start to increase. So let me use another chalk, maybe a blue chalk, to redraw this in a little more cleanly and show you what the trajectory can then look like. So we had this initial bistable system. These were the stable states. What I'm saying the trajectory will do, let's say you start somewhere here, you will very quickly go there, you'll sort of creep along there, you'll fall down, creep along here, fall off this edge, jump here, and keep going. And that's an oscillator. So it's important to emphasize here that the system we have now, a two-dimensional system which is consisting of both these equations, is not a bistable system anymore. We started with a bistable system if you only took the first equation and you had B constant. But once you make B depend on A in this way, then it's no longer a bistable system. It has some hint of the original bistability because you're spending a lot of time. Remember, this goes very, very quickly, and this goes really slowly. So this is very slow, this is fast, and this is slow. Yeah, I'll write bigger. Can everybody see the diagram? And you see where the oscillation is happening? So remember, the arrows were very large in this direction and small in that direction because of this condition I had put, that all changes in B are very, very small and slow, rather slow, compared to changes in A, which are very quick. And therefore, in this part of the trajectory where you're going up here, this will be fast. And where you're going mostly along B, this will be slow. And this will be fast, and this will be slow. So let's see how that plays out. I mean, let's draw this as a function of time, and maybe you'll see more clearly what happens. And I'm hoping you'll start seeing the connection with the yeast very quickly. Okay, so let me start somewhere. Let me start, this is the cycle that we are in. So let me start somewhere up there. That is relatively high B, high A. So we're just going to look at A. I'm going to try and plot A versus now time. So you start high with A. You are going to go down a bit in A, but very slowly. So what's going to happen is you are somewhere high on A, you maybe drift a little bit down in that time, and you drift down by that much. And you do this rather slowly. And then suddenly there's a big drop in A, right? So over time, you'll see a large drop in A. What happens after that? Then you start having a slow increase. So you'll have a period where you'll start to grow slowly like this, and then you'll fall off that edge, and you'll jump back up. And you'll jump up here, and then a slow decrease, jump down, slow increase, jump up, slow decrease, and so on. So essentially, this is an oscillator that you've built from a bistable system, which has two time scales, and is kind of reminiscent of the yeast-like oscillations. If you can see the ones he has written on the other side, you can see that there is some similarity. There are two time scales again. There is something slow happening over 2 hours, something fast, something slow, then something fast, slow, fast. Whichever way you look at the waveform, whichever waveform you have. Any questions up to this point? I'm sort of deliberately keeping it a little bit abstract because I was doing it abstractly at this point. Until this point, I've not even made a connection with the yeast, other than the fact that I have taken some kind of idea of starting with a bistable system, wanting an oscillation with two time scales. So far, I've really not taken any of the details of the yeast system. It's just an abstract idea of how do you build an oscillator when you start from a bistable system. But the reason I went down this whole road is because I had some intuition that the kind of oscillation you would get would look a lot like that, or at least have enough features for me to be happy, for me to be excited. There's still a question of how do we actually translate all this into the yeast scenario? How do we, does it make sense over there? And what can such a model tell you about the biology? And at that point, we'll have to go back and I'll have to start talking to Sunil again. So exactly. I mean, this is a very good question. There are many, many ways you can make an oscillator. And there are many ways you can make an oscillator of two time scales. I said that this is a subset of relaxation oscillators. Relaxation oscillators in general often do have two time scales. And relaxation oscillators are not always built on a bistable system. So why this model? Why this model really was pretty much precisely following the following train of thought. I had heard a talk and had a discussion with Sunil where he said, I have a bistable system. Somehow it is bistable, and yet it also oscillates. These cells can be in two states in some scenarios, but in some other scenario, when I put it into a chemostat, I can get an oscillator out of it. So part of the reason for going down this line rather than another line was to, in fact, try and see, can I start with a bistable system and make an oscillator out of it, which has 2 time scales. The 2 time scales was something I pulled out as a feature I wanted to make sure that whatever I'm looking at. In some sense, at this point, it's really a shot in the dark. There is really no reason to believe that this model, rather than another model of oscillations that could give the same shape, would be more or less useful. It really is. It's just something that connected with some other work that I was doing, and therefore, it was a direction I was happy to explore. And it's really a shot in the dark. It's very important now to sit back and ask, okay, so in the yeast scenario, if we now try to put in some of the details of how yeast works and translate this model into a little bit closer to the yeast thing, is there anything sensible that could come out which would actually be useful for Sunil or me or whatever, something that would tell us about the biology of the system, something that would tell us about new experiments, new questions we could ask. This is very much the next question, and it could have failed. And I will try and argue that it did not. But the fact that it did not was not something I could have predicted. It could very well have been a completely different kind of oscillation. Maybe, nevertheless, I would have felt this was still worthwhile to do, because even if this had turned out to be not something we were very confident about as later on as being a decent model of this oscillator, I would have found out that this is not the way to do it. And sometimes it's useful to, not just to have a model that fits and works, but also to have a model that kind of works in some ways, but doesn't work in other ways, and you learn why it doesn't work. And that gives you also a lot of information. So your point is perfectly valid in that sense. And I have nothing much more to add other than it was really just a historical accident that I had previously worked on bistable systems, and for completely other reasons was trying to make a bistable system, an oscillator, many, many years before I met Sunil. And those ideas just came back when I heard him talk. So it's a little bit of historical contingency, let's say. Yeah.
Can you hear me?
Yeah.
Yeah. So in this system, if you have one time scale, do you still get an oscillator?
No, you don't. So I'll tell you what happens. As you make these numbers closer to 1, that's easy enough to do, the oscillations become more smooth. So instead of having these very slow, fast transitions, you start smoothing out a bit. All the arrows start bending a lot more, So essentially, your trajectories will start getting more smooth, and the time scales that you spend going this way versus that way will start coming closer together. Your trajectories will start looking overall much smoother, like that. And at some point, when you get close enough to 1, you actually stop having oscillations, and your trajectory, in fact, spirals in somewhere here. And from a dynamical systems point of view, one can actually explain that. One can, so I only drew one part of the system. Let me draw this again to explain this a bit more to you. This is A, and this was something like that, right? This was dA by dt equals 0. But there's another curve which is dB by dt equals 0. And that curve can be various things, but amongst the possibilities, it could be something like that. B starts off high when A is low, and B becomes low when A is high. That's the inhibition. It turns out that the oscillations, now I'm just giving you some mathematical details. You can read about this in the paper. It's up on the website in the reading materials. So I'll just tell you the results without proving anything. That the oscillations can only happen if this line goes through the dotted line part, and you can prove this, and the time scale is very different. So if these numbers are much smaller than 1. In that case, this point, which is the only point where you can have a steady state potentially, is unstable. As the time scale gets closer to 1, this becomes stable. And once this becomes stable, essentially all the trajectories get attracted to this and you no longer have an oscillator. You only have a steady state. Okay? So those are a little more mathematical statements one can make. One can also say things like, suppose I used parameters that did not make this curve go nicely through. It's easy enough. I could have changed the K, or I could have changed the sigma in such a way that it went like that. And it did not cut here, but it cut here. In that case, one can say that this steady state, it's also the only one steady state that's around, but this steady state will be stable, and there won't be any oscillations. So as you change this, and this is a point we'll come back to with the yeast case, as you change parameters here, as you change parameters of its inhibition, maybe this value or even the Hill coefficient, you can actually destroy the oscillations. And you can get a situation where the oscillations break down and stop. And when we talk about the yeast, I'll actually try and relate that to experiments Sunil could do. Yeah.
There's no bistability now once the oscillation.
There's no bistability now.
So there's only a limit cycle which is going on. And secondly, the autocatalysis is very important. If you don't have that, you don't get oscillations.
Yes.
And
so both things are important. In some sense, I very much started with a bistable system and I added a negative feedback loop. Loosely speaking, one can make statements like the following. And let me just make these statements out so that you know of them, and we can discuss these in more detail. There is a positive feedback loop here. There is a negative feedback loop here. Fair enough? Everybody knows what those terms mean? Roughly speaking, there is a loop. A loop is simply that one variable maybe affects some other variables or not, but comes back to itself. So A affects itself, and A affects itself via B. And if you just count the number of inhibitions, if there are an odd number of inhibitions around a loop, roughly speaking, that's a negative feedback loop. If there are an even number or no inhibitions, then that's a positive feedback loop. You can make rather general statements for a certain class of dynamical systems where you can say that a positive feedback loop is a necessary condition for bistability. If you have no positive feedback loops, you certainly cannot get bistability, but it's not a sufficient condition. So you might have a positive feedback loop and not have bistability. For oscillations, you make the opposite statement. A negative feedback loop is necessary for oscillations, but it may not be sufficient. So you need to have a negative feedback loop somewhere to get an oscillator, but you need more ingredients. It's not sufficient. It turns out that very often to get a nice, robust oscillator, there are many, many cases of oscillators, even within biological systems, where a combination of a positive and a negative feedback loop really gives a very nice oscillator. This is one such example. But this is a particular example where the positive feedback loop was, let's say, in some sense so strong that it actually produced a bistability on its own when you didn't have the negative feedback loop. And that produced an oscillator when you added the negative feedback. You can also get oscillations if the underlying system is not bistable. So there are systems where you'll have a combination of positive and negative feedback and you'll get oscillations. But there's no real underlying bistability if you break the negative feedback loop. I think Albert Goldbeater will actually talk about quite a lot of these systems. Many of the circadian systems are like that, where you have a combination of a positive and a negative feedback loop, and they give oscillators. Does that more or less answer what you wanted? Okay. No? Yeah.
And when you couple with another model, you get bistability?
You start off with oscillators, and then when you add some process, you get bistability. Yeah, I'm trying to think if I know of anything off the top of my head. I'm pretty sure you can construct something like this. If nothing else, you could construct something that is bistable between two different oscillations. So you could have two oscillating trajectories or attractors coexisting. So that's a bistability of a different sort. It's not bistability of two stable steady states, but you could have two stable oscillators or limit cycles. That certainly you can get. So yeah, let me think a bit. If I can think of a simple way in which you could take an oscillator and make a bistable system out of it, I'll tell you. But I'm pretty sure you could do that too.
I had another question. So what are the constraints on how you build that equation between B and A? In the sense that if you make B independent of A, is B also following a bistable? Can B also be constituted?
So the way I've written it, certainly not. You could make B a bistable system also, and I have not. So I'll tell you what. Let's think of the B equation as essentially giving some line. It may or may not be this line. It could be some other line. The mathematical result is actually rather simple. You will get an oscillator provided whatever is the B line, the B, dB by dt equals 0 curve, as long as that passes through this and only through that region. So if I were to think of a bistable version of B, you would have something that would do this, something that would do that, and then maybe something like that. And I have a feeling that would also work in its own way. So as long as you set it up so that you were, this is an intuitive answer, I'm not betting my life on it, but it feels to me like this would work. Yeah, I do think it should work. But you would need to set up the parameters so that they intersect in the right way. I mean, maybe I can give you very quickly, maybe it's useful at this point to give at least an intuitive idea of why this rule that you should go through like this. And it's simply the fact that, let's compare it with the other case which I drawn like this. It's simply the fact that you can't overshoot. So over here, what would happen if it was going through? This is the steady state. Wherever you started, I mean, let's say you started here, you would jump to this point very quickly, and then you would start creeping along this, right? And as you creep, you would get closer and closer to the fixed point, and you'd hit it. There's no way to really avoid that fixed point. And similarly, if you were starting somewhere, so I'm talking about this system, let's not confuse this, right? We're talking about this system. Suppose I was starting here. Very quickly I'd jump down, then I'd creep along here, and I would fall off the edge, come here, but once I'm creeping along this edge, I'm going to hit that point. And therefore, you can't really keep oscillating. You would always hit the fixed point, somewhere or the other. You might take a while to hit it, you might go around a loop once, but eventually you would hit that point. The only way to really avoid hitting the fixed point, the steady state, not necessarily stable, but the steady state is to have a possibility to overshoot and then overshoot it again. And that's what happens when you are going like this. Here, you'll creep along and you will overshoot. Here's where the steady state is. You will get past it. And as you creep along, you will overshoot because you're nowhere near the fixed point. And once you overshoot, you'll fall down and then keep going. So by having this go through, you have the possibility of repeatedly overshooting a steady state that potentially is there, but you never come close enough to it to be kind of trapped. That's intuitively. Mathematically, one can do the kinds of things I was telling you about yesterday of actually investigating the stability of these steady states and showing that it's actually unstable, and therefore you can also go on to show that there must be an oscillator. Yeah.
I was thinking, can you hear me? I was thinking of Emilio's comment. There's an interesting way in which you could do that, which is to have a stable state separated from a limit cycle by an anti-limit cycle.
Yes.
And if you could do it in two different directions, you could go from the anti-limit cycle to one of the two stable states.
There are systems where you can have a limit cycle and a stable steady state separated by an unstable limit cycle somewhere in between. This works better, maybe the only comment I have to add is it works better in 2 dimensions. In 2 dimensions, you can see that an unstable periodic orbit can kind of separate space into 2 parts, which cannot interact with each other. And inside you could have maybe another limit cycle or a stable state, and outside you could have something else, a stable state or a limit cycle. But in 3 dimensions, things can be more complicated because a single orbit cannot separate space into 2, an inside and an outside. And therefore, there might be ways that things will be complicated. So in 2 dimensions, it probably works very well. I can repeat the question.
So seems that A and B, they are like bulk properties of the whole culture, but how can you connect them to like individual events?
Excellent. This is a very good question, and it's precisely what we will have to do when we start to draw this in terms of yeast. What she's saying is that a lot of the time, until now, all of the bistability we've been talking about has been at the level of a single cell. It's some gene network inside a cell. Even the yeast thing, it probably is some kind of, two different, some genetic network that has some underlying bistability. And if you're in one state, you're proliferating, in the other, you're quiescent. But the kind of oscillator we are talking about in the yeast is not that. It's not one cell, it's a whole population. And what is changing from there to there is the number of cells in the Q state, the number of cells in the G state. So if we're going to use this idea to build an oscillator based on a bistable system, we need the bistable system, we need this to not be something about one cell, but to be a quantity like the number of quiescent cells in the population, right? That's what I want this to be. So the immediate analogy when I go to yeast will be something like the fraction of Q cells to the total number of cells. So how do we make that leap? We will have to make a leap, and that's what I will do. Well, I don't know if it makes sense to do it now, or should I take a break now? And Vidya, what do you think? A break now. So when we come back, I will come immediately to what you're talking about, and we will try to take these ideas and immediately put it in the context of a population of yeast and ask, how do you get a bistability in a population? So you know that cells can take 2 states, fine. Let's assume that's the case. How can we get something where, given all other parameters in the chemostat constant, the rate of feeding, the rate of dilution, whatever, if we keep those constant, can you still get a bistability? A bistability, let me just leave you with a thought. It would mean in that case, in a population case, that there are 2 possible states the whole population can be in. There's a state that a population can be in where, say, 90% or 95, 99% of the cells are quiescent, and that's stable. And there's another state for the same parameters, the same feeding rate, the same volume, the same density of the cell culture, where that same population could be in a state where only 65% of the cells are quiescent. That's our immediate next task. If we can do that, then we can translate all these ideas to make an oscillator in the number of cells that are quiescent. And that's what I'll try to do after the break. So we still have a little bit of time. If there are more questions, please feel free. Yeah. Yes. Yes. Here, you are somehow, so it's indeed true that at A, there are sort of 2 arrows coming in. And there is an effect of B and an effect of A. And in this particular case, I have made it such that there's a common function that is a function of A and B. But he's absolutely right. You're absolutely right. You do need that. In a sense, I built all this on the idea that actually there was some parameter alpha, which was a parameter. So maybe, the best way to draw this is not like this. Maybe a good way to draw this is to say there was a parameter alpha that expressed some feature of this interaction. And what this is doing is affecting that parameter. So that perhaps is a more accurate way of representing this fact. He's absolutely right that you need somehow B and A to be acting together in order to get this effect. All right, then maybe we stop now and come back.
Thank you, sir.